Welcome to the debate. Imagine you completely change your diet today, like you go totally vegan, or maybe you switch to a strict carnivore diet.
Right.
Within just 24 hours, an entirely new, almost alien ecosystem springs to life inside your gut. Microbes that were starving suddenly multiply by the billions, and the ones you used to feed simply die off.
They just vanish.
Exactly. So looking at it that way, it seems like the food on our plate controls everything about our microbiome. But what if your DNA has already built an invisible microscopic fence? One that dictates exactly which of those bacteria are even allowed to survive in the first place. I mean, what if you aren't the one making the rules at all?
That is, well, it's the ultimate biological standoff. We are looking at the modern microscopic version of the classic nature versus nurture debate. We have this auxiliary organ made up of trillions of bacteria, archaea, fungi, viruses.
A whole universe in there, really.
Yeah. So is this ecosystem a highly malleable community shaped almost entirely by environmental plasticity? Meaning, the sheer volume of what we feed it? Or is this ecosystem strictly bounded by the structural, metabolic, and immunological constraints hardwired right into our host genetics?
Well, I will be taking the nurture side of this. I am going to argue that the environment, which is overwhelmingly driven by our dietary intake, is the dominant driver here. Diet provides immense environmental plasticity. It is this daily, massive external input that dictates the microbiome's real-time state. Essentially, your gut ecology is a direct, highly malleable reflection of what you consume.
And I am taking the nature side. I'll argue that host genetics establish strict boundary conditions. Through specific genetic loci, the host genome erects an active mucosal firewall and secretes an endogenous nutrient landscape that effectively dictates which microbes are even permitted to set up shop. The host sets a definitive, unyielding ceiling on how much your dietary interventions can actually alter the landscape.
Let's start with the sheer weight of population-level evidence because, honestly, the epidemiology paints a very clear picture. The exposome, meaning our environmental exposures, completely overrides genetic ancestry.
Okay, but
Wait, let me just give the data on this. We have massive human cohorts showing that genetic ancestry exerts a minimal, often statistically insignificant, baseline effect on overarching beta diversity. And for anyone listening, beta diversity is basically just a metric we use to measure how different my gut's microscopic neighborhood is from yours.
Right, it's the overarching structural comparison between two different communities.
Exactly that. Take the pivotal Israeli cohort study. They analyzed over 1,000 healthy individuals from highly diverse ancestral origins who all shared a relatively homogeneous environment in Israel. They found absolutely no significant correlation between genetic ancestry and microbiome composition.
None?
None. Instead, genetically completely unrelated individuals who simply shared a household exhibited profound similarities in their microbiomes. More than 20% of the variance in their gut neighborhoods could be directly inferred from environmental factors related to diet and lifestyle. But the average global heritability, the genetic influence, was only 1.9%.
Look, I hear those numbers, but global beta diversity metrics are a remarkably blunt instrument. By looking at the overarching neighborhood, you are zooming out so far that you obscure the highly specialized, localized genetic control happening on a micro scale. I mean, the gut microbiome is not just a blank slate waiting for your lunch to dictate its form.
But how can you dismiss the numbers? I mean, the Twins UK registry, which is essentially the gold standard for parsing genetics from the environment, corroborated that Israeli study. They capped the global genetic heritability strictly between 1.9% and 8.1%. And when we look at intervention studies, shifting a human from a plant-based and an animal-based diet profoundly alters the community structure in just 1 to 2 days. Diet acts instantly and it acts broadly.
I am not saying diet doesn't act fast, but you are treating all microbial taxa as if they are equally important to host physiology, which simply isn't true. Certain critical keystone microbes are consistently heritable. Look at the Christensenelliaceae family of bacteria.
Okay.
It has a heritability of up to 40%. It is intrinsically linked to the host's body mass index and a lean phenotype. The host actively preconditions the gut environment for this specific family. So diet merely feeds what the host has already allowed to live there.
Let's stop right there because this brings up our first major clash, right? Population level homogenization versus what you're calling highly heritable hubs. If we look at the Dutch Microbiome Project, they profiled over 8,000 individuals and confirmed that shared environments, co-housing, and even socioeconomic status shape the microbiome far more than genetics. So if global heritability averages a mere, say, 5%, shouldn't we view your Kristen Salacey example as a minor localized anomaly rather than a foundational boundary?
Not at all.
Let me offer an analogy. If a house is 95% built out of wood, which we can equate to diet, and only 5% built out of steel, representing genetics, we call it a wooden house. Diet is the material that actually builds the structure.
It is a neat analogy, but I just don't buy the conclusion. That 5% steel, that represents the load-bearing pillars of the house. Without those pillars, the wood is just a pile of timber. I acknowledge the broad, rapid impact of diet, but you cannot dismiss the 5% when it dictates the entire structural integrity of the ecosystem.
Structurally, how, though?
Well, that Kristen Salacey family I mentioned, it doesn't just sit there acting as an isolated anomaly. It forms a highly heritable synthropic consortium with archaea, most notably a microbe called Methanobrevacter smithii.
Okay, for the sake of the listener, break down what a synthropic consortium actually means in this context. Why does that specific interaction elevate it to a quote-unquote load-bearing pillar?
Right, so a syntrophic consortium is essentially a microscopic buddy cop system where one organism's trash is another's absolute treasure. It defines a fundamental, almost cutthroat interspecies metabolic dependency. Members of the Kristen Salacey family efficiently ferment simple sugars, and as a byproduct of eating those sugars, they produce substantial amounts of hydrogen gas. Now, if that hydrogen builds up in the gut, it causes thermodynamic inhibition.
Meaning they basically choke on their own exhaust fumes.
Right, exactly. The chemical pressure gets too high and the fermentation process just grinds to a halt. This is where Methanobrevacter smithii comes in. It specifically utilizes that exact hydrogen waste as a substrate to reduce carbon dioxide into methane.
Oh, wow.
By continuously scavenging the hydrogen gas, the archaea prevent that thermodynamic inhibition. They keep the engine running, facilitating the optimal growth of Kristen Salacey.
Hold on, you're losing me a bit here. Are you saying the host genes purposefully set up this specific biological loop?
Yes, that is exactly the point. The host genetic predisposition successfully selects for and maintains this specific loop, completely resisting the environmental homogenization you see in those broad cohort studies. And what is critical here is that this specific heritable consortium fundamentally alters host adiposity, how we store fat.
Really?
Yeah. The host's genetic predisposition to harbor this network is inversely correlated with host BMI and metabolic disease risk. If you introduce these specific microbes into germ-free mice, it curtails their weight gain entirely independent of their caloric intake. This means genetics dictates the most consequential keystone species. The steel pillars define the shape of the house, regardless of how much wood you stack around them.
I see the logic, but I'm going to push back on the scale of it. If host genetics are supposedly controlling these keystone species, how can we possibly ignore the sheer volume and chemical potency of external dietary inputs? Every single day, we dump massive amounts of food into the system that completely overwhelm the gut's metabolic state. Let's look at exogenous nutrients, the food we eat, versus endogenous nutrients, what the body produces itself. The plasticity of the microbiome is mediated by the host's metabolic response to food. I want to bring in the Bilophila wadsworthia model.
Oh, right, you're bringing up the sulfite-reducing pathobiont, a microbe that's usually harmless but turns rogue under the right conditions.
Precisely. Let's say someone eats a diet really rich in animal-derived saturated fats. Think of a heavy ribeye steak, a typical Western diet staple.
Sure.
That specific food intake uniquely alters how the liver packages bile acids. The liver shifts its production towards something called taurocholic acid. In simple terms, your liver creates a very specific sulfur-rich type of bile just to break down that animal fat. When that specific bile reaches the lower intestines, it acts like a highly specific toxic energy drink. Bilophila wadsworthia utilizes a specialized enzyme to rip the taurine off that bile salt, and it uses it to breathe in an environment with no oxygen.
And the byproduct of that respiration is hydrogen sulfide.
Copious quantities of hydrogen sulfide, which is essentially toxic gas. It severely compromises the intestinal epithelial barrier. It causes a leaky gut. It triggers a massive inflammatory immune response, and it exacerbates metabolic dysfunctions like fatty liver disease and insulin resistance. The Bilophila story unequivocally demonstrates that diet rapidly creates a very specific ecological niche. The food you ate forced the liver to make a specific bile, which allowed a rare, dangerous microbe to bloom and dictate your health outcomes. Diet overrides whatever genetic baseline you had that morning.
Okay, I'll give you the bilophile example, but you're missing the bigger picture here. You are entirely focused on the food on our plate. What about the food our own bodies produce?
What do you mean?
You act as if the microbes are fasting between meals. The host is always feeding the microbiome, completely independent of what you eat. Host genetics exert profound control through the continuous, endogenous provision of specific carbohydrates.
Oh, you mean the mucosal glycans? The sugars lining our intestinal walls?
Yes, exactly. Consider the fucosyl transferase 2 or the FUT2 gene. This gene controls the addition of specific fucose sugars to our gastrointestinal secretions. Individuals who have a functional FUT2 gene are called secretors. For these people, the body is essentially sprinkling sugar along the intestinal wall. It serves a critical dual ecological purpose. It gives commensal bacteria physical attachment sites to hold onto, and it provides a constant, diet-independent supply of fucose as a carbon energy source.
But again, isn't that just a baseline? Once you introduce a massive influx of dietary carbohydrates, say a huge bowl of pasta, doesn't that just completely swamp the tiny amount of mucosal fucose the body is secreting?
No, because it applies intense evolutionary selective pressure on the microbial community over a lifetime. Specific bacteria, notably within the Bifidobacterium genus, possess specialized enzymes that have evolved specifically to harvest these host-derived fucose sugars. And here is where it directly impacts the listener. 20 to 40% of the global population have mutations in their FUT2 gene that make it non-functional. They are non-secretors.
Right. They don't produce those sugars.
Right. Metagenomic analysis show that non-secretors experience a fundamental genome-driven reprogramming of their gut's energy metabolism. Their microbiome is forced to adapt to the complete absence of mucosal fucose, which leads to broad-based depletions in pathways that synthesize vital amino acids and vitamins. This happens regardless of what they eat. The host biochemistry forces the microbiome to adapt before a single bite of food is even considered.
Okay, let's take a step back. I understand the FUT2 mechanism, but let me give you a different perspective on how genes and diet actually interact. You're basically talking about gene by environment, or GXE interactions. And if we were going to talk about GXE, we have to look at the LCT locust, which encodes that digestive enzyme lactase. This is arguably the most statistically robust genetic association with the gut microbiome we've ever found.
Because it perfectly illustrates the genetic ceiling I'm talking about.
I completely disagree. It illustrates that the genetic ceiling only matters when diet activates it. Let's break this down for anyone listening who is lactose intolerant. In individuals who are lactase non-persisters, meaning they lack the genetic mutation that allows them to digest dairy into adulthood, when they drink a glass of milk, that dietary dairy bypasses absorption in their small intestine. It travels down into the large intestine, alters the fluid balance, and serves as an exclusive massive feast for specific fermenting bacteria.
Right.
Dietary dairy causes a profound explosion of Bifidobacterium species. but only in those lactose-intolerant individuals. The genetic predisposition only restructures the microbiome if the environmental trigger, the milk, is introduced. Without the diet, the gene is ecologically silent.
But the gene provides the permission for the diet to act. It dictates exactly how that environmental input will be metabolically partitioned between the human and the bacteria. If you have the gene to digest milk, you get the calories. If you don't, the microbes get the calories. And we see this permission structure even more rigidly when we look at the mucosal firewall, the immune system. The immune system doesn't passively tolerate the microbiome, it actively engineers it.
You are talking about the innate and adaptive immune filtering.
Exactly. Let's look at the innate immune system. We have the NOD2 receptor. Think of it as a microscopic alarm system inside specialized cells in the gut called Paneth cells. When this receptor detects structural components of bacterial cell walls, it rings the alarm, stimulating the production of potent antimicrobial peptides. It's the body's natural bleach.
Right, keeping things in check.
Now, if someone has a mutation in the NOD2 gene, those Paneth cells fail to detect bacterial encroachment. The bouncer falls asleep at the door. The result is a profound ecological vacuum. Pathogenic microbes expand completely unchecked, leading to severe dysbiosis, like we see in Crohn's disease. The microbiota fundamentally destabilizes the host, and it frankly does not matter whether that patient is eating broccoli or bacon.
I mean, I don't disagree that severe genetic mutations in immune receptors cause devastating dysbiosis. That's clinical pathology. But what about the healthy state?
Even in the healthy state, the adaptive immune system is constantly, aggressively curating the ecology. Your body pumps 2 to 3 grams of secretory IgA into the intestinal tract every single day. It continuously samples the environment and physically coats between 10 and 50% of your gut bacteria. And it isn't just killing them. Secretory IgA is like putting a leash on a dog. It alters bacterial gene expression, down-regulating their virulence factors and forcing them into a metabolic symbiosis. The genetically programmed immune repertoire actively decides which microbes are tolerated and which are suppressed.
Okay, I accept the reality of the gene-environment interaction and the immune firewall. You've made that clear, but let me introduce a specific framing to synthesize this, because I think it bridges our gap. If the microbiome is an ecosystem, genetics is the geography, the mountains, the valleys, the altitude of the terrain. But diet is the weather.
Okay, I'm listening.
The geography defines the absolute physical boundaries. A mountain won't suddenly turn into an ocean just because it rains, but the weather, the diet, dictates the dynamic day-to-day conditions. It determines if there is a flash flood, a devastating drought, or a massive spring bloom. While the geography defines the boundaries, the weather is what actually animates the ecosystem in real time.
I actually really like your analogy, but you're entirely forgetting what happens after a storm. If genetics is the geography and diet is the weather, we have to talk about where the water settles when the rain stops. We have to talk about colonization resistance and the homeostatic set point.
Meaning the ecosystem's ability to bounce back to its original state.
Precisely. A healthy gut microbiome functions as a highly resilient ecosystem. Because of that structural and immunological geography, the microbiome has a genetically defined homeostatic set point. When you introduce a drastic dietary intervention, you create a superficial storm. Yes, the microbiome composition shifts rapidly. But because of intense nutrient competition between species, the physical space already occupied in the mucus, and the host's immune adaptation, those changes are overwhelmingly transient.
So you are arguing that once the dietary pressure is removed, the ecosystem just snaps back to the genetic baseline.
It snaps right back. And this is the exact reason why that expensive probiotic pill you bought might work wonders for your friend, but does absolutely nothing for you. Studies using invasive endoscopic sampling show that the human gut lining poses a highly individualized, personalized barrier to microbes trying to move in. Some individuals naturally have a permissive gut, but others have a resistant phenotype. In resistant individuals, the genetic terrain actively repels the probiotic strains. The new bugs never establish a persistent foothold. The geography dictates the absolute limits of the weather's impact. Unless you apply constant, indefinite dietary pressure to overpower those established networks, the genetic ceiling holds firm.
I think we're finally hitting the core of the issue here. Look, I will readily concede that the microbiome cannot be treated as a purely malleable, blank slate reflection of our dinner plate. Host DNA absolutely provides the infrastructural geography, the endogenous fucose sugars you mentioned, the digestive enzymes like lactase, and those strict immunological bouncers at the door.
Right.
However, I maintain my position that diet remains the most potent, immediate, and crucially, the most modifiable tool we possess for shaping our metabolic reality. The fact that shifting your diet can cause rapid blooms of organisms that either produce toxic gases like hydrogen sulfide or beneficial compounds that heal the gut lining proves that environmental plasticity is the proximate driver of our health outcomes. The weather might eventually pass, but a severe enough storm permanently alters the landscape.
And I will acknowledge that dietary inputs are the fundamental fuel of the system. We can't ignore the raw caloric intake. Diet introduces the chemical energy that drives those short-term compositional shifts, and it serves as the environmental trigger that activates pathways like the lactase gene. But my point remains. We cannot escape our biology. Every dietary intervention, every restrictive eating plan, every probiotic capsule is ultimately tethered to the unyielding immunological and structural thresholds set by your genome. You simply cannot permanently remodel the ecosystem without the host's genetic permission.
Well, it seems we have arrived at a fascinating convergence. Treating this as a binary nature versus nurture dichotomy is fundamentally flawed when applied to the microbiome. It is a brilliant, intricate dance of gene-by-environment interactions. The future of precision nutrition and therapeutics cannot rely on universal dietary recommendations like, everyone should eat this specific fiber. It must be mapped to the individual's unique biological constraints.
Absolutely. It is the hologenome theory in action. The human host and the microbiome are not separate entities. They operate as a single, integrated functional unit.
As we wrap up today, we leave you, the listener, to consider the incredible complexity of your own internal ecosystem. The next time you sit down for a meal, remember that you are not just feeding yourself. You are casting complex weather patterns across a vast, microscopic landscape, a landscape whose deep valleys and high peaks were carved by your ancestors' DNA. The material we drew from today offers even more depth on how individualized therapeutic interventions might one day allow us to perfectly match your daily weather to your permanent geography. Thank you for joining us on The Debate.